Hello my dear student, I am Eduhelper. Today I will take a class about biological activities. You know that in our previous class already I have discussed about bioenergetics, photosynthesis and respiration. Today I am going to describe biological activities for practical and creative questions. Hope you will enjoy this video, and after watching this video, if you have any question, please write down on comments box at YouTube segment. Thank you so much for watching this video. Okay, my dear student, now I am going to describe first experiment. Experiment name is show the necessity of light in the process of photosynthesis. Few materials are required to perform this kind of individual activity. First of all, a potted plant with green leaves, black peppers, 95% ethyl alcohol, 1% iodine solution, paper clips, water, petri dish, test tube, beaker, Bunsen burner, sprit lamp or dropper and forceps. So now I am going to procedure. You know that the potted plant should be kept in a dark place for 48 hours so that the leaves become starless. A part of a leaf on the potted plant kept in the dark needs to be covered with a pitch of black pepper and clipped in a such way that no sunlight can reach it. Okay, this is a very crucial point. Then the potted plant should be kept in sunlight for 6 to 7 hours. After 6 to 7 hours, the leaf plucked off and black tape removed and will be boiled in water for several minutes. Okay, so this is a figure. Then in order to make it chlorophyll free, the leaf should be again boiled in 95% ethyl alcohol until it becomes faded. Okay, now the leaf is to be washed in the water and drained with the iodine solution. In order to avoid any accident while boiling in alcohol, the test tube containing alcohol and the leaf should be heated inside water in a beaker instead of giving it any direct heat. Okay, this is a very much important portion, I think. Observation. So how will you understand that necessity of light? When the leaf picked up from the iodine solution, we can see the except the covered part, the remaining part of the leaf becomes blue, deep purple or black. This is a very important point. Okay, so I want to say again, we'll see that except the covered part, the remaining part of the leaf become blue, deep purple or black because of the chemical reaction between stars and iodine. Stars will turn blue, deep purple or black as sunlight cannot reach to the part of the leaf covered with the black pepper. Stars cannot be formed there through photosynthesis. As stars is not formed in that part of the leaf, it cannot turn blue, deep purple or black after reaction with the iodine solution, since sunlight has fallen on the remaining part of the leaf. Stars was produced there. It proves that light is essential for the formation of stars during photosynthesis. Okay, so there has few precautions we should follow it before performing the experiment. The potted plant should be kept in the dark for some times, at least 48 hours, and black pepper should be of such quality that no light can pass through it, And number 3, before the test, the potted plant should be kept in the sunlight at least for 6 to 7 hours. And lastly, while boiling the leaf into alcohol, it is better not to use direct heat, which I have described previously. Okay, my dear student, now I am going to describe second activity. This experiment also important for creative. You can say not only important creative but also practical. So experiment name is to show the necessity of chlorophyll in the process of photosynthesis. Okay, so again I'm repeating the name of activity: experiment to show the necessity of chlorophyll in the process of photosynthesis. Okay, here some green leaf, many hot leaf, alcohol, iodine solution, water, petri dish, test tube, Bunsen burner, dropper, beaker. These are essentials is needed to perform this kind of activity. Procedure very easy. A many hot leaf is to be picked at noon and its green portion marked. Okay, after boiling the leaf for a few minutes in water, it is then be boiled in alcohol until its colors has faded. Now, after washing the leaf in water, dip in it iodine solution in order to avoid accident while boiling in the alcohol. The test tube containing the alcohol and the leaf should be heated in water in a beaker instead of applying direct heat. Okay, so after that observation, only the green portion of the leaf become blue. This is an important point. Again, I want to repeat it. Only the green portion of leaf become blue, deep purple or black after the leaf form iodine solution. So why? Because of only the green portion of the leaf produced carbohydrate through photosynthesis because of the presence of chlorophyll. Since there is no chlorophyll in the non-green portion, carbohydrate was not produced. Because of the carbohydrate, the green portion becomes blue or deep purple or black in the iodine solution. So I want to mention one precaution: while boiling the leaf in alcohol, it is better not to use direct heat. Okay, so the common precaution again. So I hope so you have understood second activities. Now I am going to number last activities related with respiration. This experiment is very much important. Dhaka board already made a question depend on this experiment, so I think you should read it carefully. Okay, so experiment to demonstrate the release of energy in the form of heat during respiration, and you have to perform it at your school lab also. So essentials are 2 thermoflask, okay, 2 thermometers, 2 rubber cork with holes, some germinated peaks, 100% mercury chloride. These are essential. So now I am going to describe procedure. Label one of the thermoflask A and other as B. Some germinated chickpeas along with some water are placed in thermoflask. Okay, thermoflask A thermometer should be inserted through the hole of a cork on the flask A with it opening sealed well. It is a very important point. The remaining chickpea will be placed into thermoflask B after soaking in 100% mercury chloride solution for 10 minutes. Okay, A thermometer will also be inserted in the thermoflux B. Now after taking the initial temperature of each thermometer A and B, they should be kept at rest. So what will you see? The temperature of thermoflux containing germinated seeds marked A rises. On the other hand, there is no temperature change in the other thermoflux B. No change here. Okay. So to sum up, you can say that the P's in the flux A carried out respiration and release of energy in the form of heat. So the temperature in the flux rises. But in thermoflux B, as the seeds are dead and sterilized because they are soaked in mercury chloride solution, the respiration process does not occur. So the temperature remain unchanged. Very easy. So there has a few more precautions. The seed should be fresh and well germinated, and secondly, the tip of the thermometer should be positioned at the middle of the stacks of seed. So hope you have understood this experiment. If you have any question about these experiments, you can write at comment box of YouTube segment, and I will try to give your answer as soon as possible.